do that out of equilibrium. And how can the cell bring the state out of equilibrium? One way could be, for instance, when there is a response to an external signal, a transcription factor which responds to an hormone, for instance, you have a relaxation toward a new equilibrium. And this will correspond to a nucleation of a foci, for instance, under the microscope. Another situation in which is where you have a motor, and the motor can keep driving the system out of equilibrium. And in this case, you will also generate states which are not allowed normally in a polymer. So if you want to model this kind of processes, the first one would be like a relaxation toward a new minima in some kind of potential field. The second one would be some kind of limited cycle in a dynamical system. But we have to consider that here is chromatin, so it's a polymer. So you have also energy dissipation. And my work is exactly to study the formation of this kind of state in this context on the polymer. And a model in which I am very interested and I'm studying in Coulomb lab is the model of TADS. So TADS are very interesting because they've been shown in data to be a principal factor contributing to chromatin conformation in a mammal cell. Like this region where you have increased number of contact respect to the rest of the chromatin. And now they have been studying also with more complex technique, for instance, oligopaint, which allow to see these TADs in individual cell. And in fact, what they show is that they can see it as a mean. And finally, we want in the lab of Coulomb to study the dynamics of these TADs. So to use foci tracking to study their formation and their dynamics. And these need to be inserted in some kind of theory. And the standard theory is loop extrusion. So loop extrusion is performed by Cohesin, which is a motor. The motor binds to chromatin, and then it consumes energy to extrude the loops, which start very little, and they get bigger and bigger until it finds a roadblock, which can be CTCF, can be another Cohesin maybe. And at this point, it will relax. And relaxation, in fact, is quite important, as you can see in this kind of video made by the Mirny group. Like there is memory because it's a polymer. So this can give some kind of conformational memory, which can have a very important function, also considering the fact that you cannot have loop extrusion everywhere all the time in all cell. And so this talk, I will present my insight and my modeling of loop extrusion with this idea to understand the effects of this process on the conformational memory. So, we want to do this also to model experimental data, which we will produce in our lab. As I told you, we are able to label foci and to follow their dynamics. Like we can label, in this case, 2 foci, which are separated by a place which we know that there is a loop extrusion between them. And we can try to see signals of conformational memory or energy consumption, to measure the energy consumption by looking at the trajectory. But trajectory will give us this kind of trajectories, and then we need to analyze them. And we think that we can access this information by looking at relaxation time and signal of broken detail balance. And we can do this in general using higher order correlation function, like correlation function of observable at different time scales. But this is for the future. Now let's discuss a bit the models. So the current standard approach to study loop extrusion is to use molecular dynamic simulation. Like you're looking here at a video of Mirny lab, like they simulate some kind of connectivity in 1D. And they evolve this connectivity, where this is the action of Cohesin, and they look at the conformation of a single chromosome in this case. But this is a simulation, so you need to do it many different times in order to obtain some kind of mean picture like you obtain in IC data. IC data is a population measurement. So to do that, you will have to repeat the simulation hundreds of times. You need powerful hardware. You need a lot of resources, and finally you will have some information which will be numerical. Instead, we want to do one step in the direction of getting something more analytical and to understand the process more from the mathematical point of view. For this reason, we are developing what I call a semi-analytical approach. And here I will describe you basically what does it consist. So we are using a standard model of statistical physics, and the most simple one where you can have an analytical approach to a similar problem is a 1D diffusion. Like now is not loop extrusion, is one particle in 1D which can move right, left, or stay there with a certain probability. And you can simulate it like they're doing in Mirny lab, and we see that it will go right, left, randomly. And then you can do it many different times. You will make an histogram, and with the histogram, you can see the distribution. Like you start in 1 point, then you relax a bit, you can go in 3 points, relax again, and you will just spread out using diffusion. But we don't really need to simulate this process, which is extremely simple, because we know from statistical mechanics that the MSD in function of time will be linear, will be a line. And we know that this is a Gaussian process, so we can just model the variance of this Gaussian, and we can predict the histogram with even much more precision in time without having to simulate. And we have the advantage that it will be extremely fast, and we have also a full probability distribution for the result in space and time. Now, let's go toward modeling this way a polymer. So you have, the first step will be to have 2 beads which are connected by a spring. So not really a polymer, but just 2 beads connected. And then we can measure now the position in 1D, the x-axis, of the green and the blue beads. And at the beginning, they might be in 1 point, but then by doing a simulation, like the green might go right, and then the blue will follow because there is a spring. So the blue will be pulled by the green in the same direction. And this will happen again and again, and maybe the other direction, but the result is that you have a correlation. So springs make correlations, so you can still use a Gaussian with the correlation, an extra parameter, and you can calculate the analytical solution, and there will be a Gaussian, which is spreading in the position of the green and the blue beads, and it looks like a Gaussian with a correlation. And each point in this space will correspond to a specific realization of our model. So if we wanted to go to polymer, we need to make the Rouse model. So the Rouse model is many different beads, like N connected by springs. And this is the simple possible model you can have. It models basically the connectivity, the fact that it's a polymer. It's able to reproduce statics and dynamical aspects, but you are ignoring steric and volume excuse effect and topological constraint. Typically, springs can pass through each other. And if you do that, you will have exactly the same as the 2 beads, but now you are in 3 multiplied by n dimensional configurational space. So it's a very complex space in which you have again each single point corresponds to a specific polymer configuration, and all the variables will be correlated because of the spring. So you can solve this and you can calculate the green function. So basically, what I tell you is that if you start with all the polymer, the probability of a polymer in which all the monomer are in 1 point, that's extremely unlikely. But let's start in this condition. Then we let it relax. It will open. It will make a random polymer, which open up. And the nice thing is that we can use correlation to connect it to something which we can actually observe, which is the predicted distance, which is then connected to the contact probability. And you see that starting from a point, which is flat, you will make the typical probability of contact like that you see in IC data. And this is fully analytical. So now if we want to model loop extrusion, we have to add the loops. So it will not be a Rouse model anymore, but will be something slightly modified version of the Rouse model in which you add some other spring. And this at equilibrium will show up like having another point out of the diagonal, which connects to the diagonal in the same way as diagonal objects connect to each other. This is a loop. Now, let's go out of equilibrium, because finally, loop extrusion is an active process. So let's just try to break this point like this loop at time equal tau, starting from time equal 0, and then having an open polymer and see how the IC map, how this contact map or distance maps evolve. So you can have two cases, one in which this one will relax, a polymer which relax fast, or a polymer which relax much slower. And this depends typically from elasticity or viscosity. And the result will be different. Like when you have a fast relaxation, you will forget about the loop very fast. When you have a slow relaxation, you will keep the memory of the loop for much longer time. And this conformational memory is what we think can have even a functional effect. Now, a way to measure this conformational memory, which we figure out to have something which is, we can plot it, is the entropy. So we can measure the entropy of the Gaussian model. And we can see that you start with the entropy of a closed polymer, which is reduced since you have a constraint. And then you relax toward the entropy of an open polymer. And depending on the polymer dynamics, you can do it very fast or you could do it much slower. Now let's go to loop extrusion. In loop extrusion, there is still a network model, like this kind of graph, but now the connectivity evolves in time. And it evolves according to this loop extrusion model. So again, you can have faster or slower relaxation, or you can have slower or faster extrusion. The two things are exactly equivalent. But the point is that we are looking at how the model performs in function of the velocity of extrusion. And you see that here out of equilibrium effects are clearly observable. Like when the extrusion is low, you can see at equilibrium picture all the time. When it's faster, you can see the appearance of more different phenomenology and the memory. And this conformational memory is exactly what we want to study. Now we can study the entropy, and we can see that it forms a cycle which represent the loop extrusion, but it is different at equilibrium or out of equilibrium. And we can study this more in detail. So first of all, there are three different steps of loop extrusion: when the polymer is open, when it's extruding, when it's closed. So when the polymer is extruding, you are making bigger constraints, so you will reduce the entropy. And you do it different. So this is open polymer, and this is closed polymer. At equilibrium, it's very simple. It goes from open to closed. Out of equilibrium, it never does that. It's always a lower entropy. So you're always exploring a smaller configurational space. And then you can clearly see that there is this overshoot effect. And then when there is relaxation, like when the polymer, when the cohesin is fixed at CTCF, you go back to the open polymer, to the closed polymer. And when the relaxation is in this side, or when the polymer is open, you go back to the open polymer, but you do it very slowly because here extrusion is faster. So finally, we identify the effect of out of equilibrium, which are this kind of overshoot when the polymer is closed, and this kind of undershoot, let's say, when the polymer is open. But this is still a quite complex physical system, because we have three different states. Each one already have some kind of entropy, characteristic entropy, which is very peculiar, and the effect of out of equilibrium and this effect, which are intrinsically out of equilibrium, they mix up. So we want to simplify the model just to study this out of equilibrium effect. And to do that, we can start by eliminating the open polymer and the closed polymer and keeping a system which is constantly extruding. Secondly, we can close this system, like making a loop, so that the cohesin will not fall out. Well, it doesn't fall out if it's not a cohesin anymore, but it's another motor, which probably doesn't exist, which is extruding the loop on one direction on one side and the other direction on the other side. So we don't call it anymore loop extrusion, but we call it loop transition. And this is just a very ideal model which we use as a tool to study the out of equilibrium effect of loop extrusion. So if you do loop transition, you can still start with a single loop, like here, and then you can transitioning faster or you can transition slower. And first of all, you will have some kind of transition, like you are going to a new equilibrium, a new steady state, and then you will see that the matrix out of equilibrium have this kind of tail, and this tail are the memory of the previous state. So finally, you will be in an out of equilibrium steady state for the entropy, and this level will depend on the velocity at which you are extruding. And finally, if extrusion is extremely slow, basically, you don't even have any effect on entropy. You are every single state of this dynamical system are equivalent. And what does it mean, a reduction in entropy? A reduction in entropy means that, like here I'm simulating, I'm not doing analytical calculation, thousands of polymer which are undergoing this transition to loop transition. So you start with an open polymer, and then due to activity, it reduces the size. So activity, in fact, is equivalent to a compaction in this case, to a reduction.
So Vittore, you have still 3 minutes.
Oh. So we can try to see to measure the breaking on the tail balance. For this, you have to move to an experiment which we can envision, and which will have 2 different colors, and we will follow their dynamic. We can measure their distance, for instance, while the loop is transitioning, and you can see that in fact we can look at the tail balance as the time reversal is symmetry breaking of their distance, and when it's at equilibrium, the distance reduces due to transitioning. It happened the same also out of equilibrium, but now the curve is skewed. So if you will invert the video, you will see it differently. And then you can also check the analytical result with simulation, and you see that it totally corresponds when you have like hundreds of realization of the system, of course. So now let's see what kind of data we would like to model in vivo, like will be contact maps. So there we need a much different connectivity. Like we need a model which model the connectivity in 1D first of all. So we are making a much more realistic system where you have 5 different parameters, will be the binding rates of CTCF, the binding rates of Cohesin, and their extrusion speed. And then you have to make a huge number of hypotheses. Like you have to make the hypothesis that the Cohesin loading is uniform, and maybe it's not. You can change all of them. That's the nice part of it. And you can change them also experimentally, probably. You can do extrusion on one side, choose side. You can hypothesize what happened upon collision. And then you can simulate these. And you can see that even there, when you connect this 1D simulation with a 3D simulation, relaxation matters a lot. Like when relaxation is fast, the 3D polyflux, like the probability of contact for each time, it's reflect more or less what you see here. While when it's low, you have memory which overlap a cycle of extrusion, and basically the fact that you have extrusion persists for a long time, also when extrusion is not there. Now you can get contact map by integrating in time these 2 pictures, and you will get that in fact it is the out of equilibrium effect that can make segregation between a system where between 2 TADs. Like if you have fast relaxation, you have loops, but there is no reason why things inside the loop might not doesn't mix. When instead, when the relaxation is low, finally you see barrier emerging out of this out of equilibrium process. And this corresponds also to the fact that we have a memory and that we have specificity. Things inside these data will meet more often than outside, and can have an important functional effects. But this is a work in process. We still didn't fit real data. That's the next step. Now it's just hypothetical and very theoretical. So to resume, we have 2 simplified model, one which is a single loops, which is repeating over and over. The other is loop transition, which is even more simple. We see out of equilibrium effect in terms of broken the tail balance. And we have a detailed model, which we still have to fit very well to experimental data, which predict the segregation between TADs. And with this, I thank the lab, Antoine Coulomb, and I welcome any question.
Thank you, Vittore. You did it perfect on time.
Okay, a bit fast.
So yeah, congratulations.
Thanks.
So who has questions or in the chat? So maybe I can start. So I'm am a bit far from, of course, I have questions from the biological point of view. At the very beginning, when you started by asking how you organize the genome, and you talk about the fact to be able to follow two dots, the green and the, so I was wondering, how did you, what is the kind of labeling that it was performed there?
So yes, this is preliminary data. In fact, what you have seen, I think, is some kind of telomeres, and so it's repeated sequences. You need some repeat sequences labeled with is GFP plus connected to a dead Cas9.
Okay, these are repeated sequences.
Yes.
So, okay,
yes.
I understand. And the other question I have is that you have seen that you have indeed a very strong effect because of the relaxation. So what would relax the chromatin in vivo?
So relaxation, in fact, what we see in vivo is that chromatin relax very slow, at least quite slow. So it's not maybe what relax, but it's what keep it quite constrained. Like I don't see it's relaxing very fast. But of course, time and temperature and the random activities transcription will relax it.
So your view is the fact that in vivo the chromatin is rather far from being relaxed, and so so you keep the memory much more.
Exactly. That's the hypothesis, the working hypothesis.
Okay, thank you.
Thank you.
So who wants to ask a question? Maybe.
Yes, I have a question. Vittore, did you compute the distribution of contacts as a function of the distance? I mean, the P of.
Yes. So this I didn't show it here because we are still not at that level. But it can change a lot in function of all the parameter, and it's clearly less than, like, if you look at the slope, you can go to values which are very slow, like less than 1. It's not. Yes.
Okay, interesting. Nice.
So there is one question from Vicky. Vicky, you can go ahead. Your microphone.
It's okay, Vicky.
So in between, Vicky, I will just ask the question from Mohamed Sebal. He's asking, is it possible to find fast relaxation in vivo?
That's an interesting question, but I think it's a question that needs to be asked more to the community because I don't know. Like if somebody have an idea, I think it's something which we can check with models and also maybe experiments.
Okay, I don't know. So Vicky, did you find your microphone?
So Geneviève?
Yes.
Yes.
Okay, can you hear me?
Yes, we do.
Okay, so it was along the same line again. So you have one experimental system. So maybe by changing the condition of culture of the cells, you could change things, or maybe by like along the talk of Emmanuel, by doing DDR and maybe having a chromatin which is more fluid, you would expect change in relaxation, but also putting cell in quiescence, maybe by changing serum and so on.
These are all good ideas. And one other approach would be to change the cohesin activity while keeping the relaxation, because here we are also always looking at relative effects. So both would work. If we slow down cohesin, might also work. And also I think that there are other things which we can do, like looking at the scaling in function of the dimension of TADs, for instance. There should be some variation in function of this.
Okay, thank you.
So Vicky didn't find her microphone, but she's writing something. She said, does your model favor two-sided motor or one-sided motor activity of SMCs?
Very good question. I don't have yet an answer. next meeting.
Okay, so.
But maybe we can find the literature already. like it's more one-sided, but okay.
Okay, so Cedric, you wanted to, he's gone. So Arash Goldar, maybe you can go ahead.
Okay, hi, thank you, it was a very nice talk actually.
Thank you.
Okay, just a question, is your model confined in a volume or it's just a
So there is no reason, like there is one computational reason why we needed to confine it because of numerical stability. But in theory, like what I'm doing now is to confine it in a very, very large harmonic potential, but it could be confined in a smaller one, and that's a possibility.
So there is no effect of this confinement on your contact frequencies?
No, because I'm keeping the confinement very, very blunt.
Okay.
That's why.
Okay. Thank you.